In this episode, we welcome Dr. Wolfram Brück, who shares his international perspectives on food and health microbiology. We'll discuss infant nutrition, the unique field of foodomics, and how it links our diet to chronic illnesses through our genes. Expect to learn about new food trends, such as sustainable proteins, and turning food waste into something good for us and the planet. Stay tuned. Dr. Wolfram Brück studied microbiology in Minnesota and Manchester. In 2003, he finished his Ph.D. with Professor Glenn Gibson on the influence of prebiotic milk peptides on infant health. He joined the Institute of Life Technologies as head of the microbiology laboratories in 2014 and is currently directing a National Competence Center for Research Application on sustainable diets and precision nutrition, which is to be submitted in April. Welcome, Dr. Brück.
It's a pleasure to be here. Thank you.
Such a pleasure having you on. So first, let's take a step back. I definitely want to get into some of the work that you're doing on the research side, but can you tell us a little bit more about your background and journey in this field of personalized nutrition? What does that mean and why this?
So I'm a traditional microbiologist, actually. So my journey into nutrition has been something that's ongoing from this really clinical based looking at diseases or acute disease actually to something that becomes more chronic. And of course, if we talk about chronic diseases, these are things that are not classically treated medically, and we have to really look at nutrition. And for me, personalized nutrition is really something that each individual reacts to different stimuli and to different nutrients differently based on the genetics. And this is something we're just starting to understand really as these new high throughput technologies come through. And this is something that's ongoing also in science as well. And we're getting more and more data with more and more novel technologies. And we can see that everybody sort of responds to things differently than we can say just everybody responds the same to the same ingredient. This is simply not true in most cases, actually, now we can see.
And I find that the topic of personalized nutrition just to be so incredibly fascinating, right? Because when you talk about personalized, you talk about variances across different groups. I can't imagine personalized nutrition to be any easier. It's much, I would think, comparatively, when you take a generalized nutrition approach, it is much more blanket, but it may or may not work for everyone. How do you sort of balance the innovation within precision nutrition and sort of general nutrition guidance and advice within this field?
Well, of course, we learn more as time goes by. And that's why, of course, some people might be concerned saying that, well, one day they're saying one thing, but the next day they're saying the opposite thing. And that's just basically, we as scientists, we're continually learning and we're improving our knowledge basically. And then, of course, we can change our minds as new information actually comes through. And we just have to adjust some things basically. And of course, general nutrition, of course, is we can generalize things certainly, of course, but then maybe some things come into more a personalized approach. And we do see these things like maybe some things get more personalized in terms of what nutrients become more important for certain groups of people, certain lifestyle, certain stage of life as well. Maybe some things change as well for those groups. So we're gaining a bit more focus on different groups, but maybe we're even going one step further, saying, well, in the future, we're saying we're looking at somebody's genes, we're looking at somebody's lifestyle, at somebody's nutritional aspect, and we say, well, maybe looking at the data we gathered from you, you should be doing this. And this is a journey that has started already, but we're getting from day to day, we're getting better and better in this, and we're improving things as we go along. And of course, in the future, as technologies become cheaper as well, maybe we'll actually go to the doctor and say, well, this is my gene, these are my samples, so urine, fecal samples, blood, or whatever. Have at it, tell me what's good. And the person, the doctor will actually, or the nutritionist especially as well, will be saying, well, based on your profile, based on the fluids you gave us, based on the information we have from you, you should be doing this.
What a more high value future that is, right? What a high value discussion that is with your provider, with your doctor in a world like that. Double clicking into that, in fact, you mentioned different stages of life as well, which is where I would like to understand a little bit more around your research. Now, it may not meet much on surface, but I know there is more to this. Can you please help us elaborate, or help us understand better yet,
Yeah,
your research regarding infant nutrition? Now, what are some of the
of
key
course.
takeaways, and why does it even matter for us to think about infant nutrition?
Well, for us, we have to imagine we start our journey actually before we are born. The health status of the mother, BMI, living nutritional aspects, everything basically surrounding the mother. And this goes into the infant. And specifically, me as a microbiologist, I would like to look at the microbiota, of course. So the guts, bacteria that are in humans as well. And we start generally what we think of at least right now when you're born, you start off with a gut that's almost sterile, as far as we know. This might change in the future again. And then based on your mode of delivery, what you're being fed, whether it's breast milk, whether it's formula, whether it's anything else, because maybe you live in an area where there's a crisis happening right now, and some of these normal options might not be available to you. Um, this decides on how you get inoculated, because we can imagine, of course, these foods contain bacteria, and these bacteria then inoculate into the gut and start to proliferate with all the foods that are being available to those organisms in the gut. And we do see that based on this programming, what bacteria come in at what stage, from what mode of nutrition, from what mode of delivery as well, we get programmed first of all with our immune system. We get programmed with different health aspects. And depending if we maybe don't get the best start in life, we get these bacteria that are maybe not as good as like bifidobacteria, lactobacillus, those organisms we really know quite well and are called probiotics, of course. Maybe then you're more programmed to some diseases like chronic disease, metabolic diseases, some immune problems, um, allergies, cardiovascular problems, or all these chronic non um, communicable diseases, you might actually be programmed already at your first days of life. And this is what we really want to look at, because my interest is the first 6 months of life, what actually happens, and what are the influence in the first 6 months of life that really start the programming of this adult, this human that you're going to become in the future, and what actually is that influence with these first few days in life, basically, and the microbiota and the nutrition and everything else surrounding all these different factors.
That is so incredibly fascinating. Tell us more about this research. What have you been able to find? And I know you've also done this study and research across the United States and Europe. Help us understand some of the similarities or even differences across countries.
Yeah, so I actually went as an exchange student to Minnesota, first of all. So I finished my high school over there and then went to the pre-med path in Minnesota. And but then basically I became more and more interested in bacteria and through different courses. So kudos to my professors back in Minnesota for interesting me in microbiology and sort of like changing my life to sort of like become from what I saw definitely becoming a doctor basically to becoming a microbiologist, and now more microbiological nutrition, maybe you could say something like that. And so I did my undergraduate over there and then went to a medical school, but looking at the medical microbiology in Manchester. So I worked with a diagnostic lab in Manchester at at the University Hospital, at medical school as well, and looked first of all in respiratory disease, because we're in the WHO Center for Pertussis, so whooping cough actually. And I saw there's a lot of troubles with infants. Of course, we try to get new methods for early detection, better detection of these diseases. But then I sort of like thought this is quite grim as well. I maybe want to start at positive aspect, how to improve situations before they actually happen. That's when I came to Reading with Glenn Gibson as well, and then learned about probiotics and prebiotics and how they can influence actually how we grow up and how we become adults, how we become, you know, a functioning organism or a functioning ecosystem, we could almost say, because we harbor, of course, trillions of different bacteria. And there we saw, of course, with probiotics and prebiotics that if you eat those organisms, we eat these different compounds that can feed the organisms in your gut or different niches as well, basically, you can actually improve the situation with this. And we can see this especially with infants. And I worked on infant formula or improving infant formula to make it more like breast milk, because breast milk, of course, is the gold standard still, what we want to feed our baby. When this is not possible, we have formula, but of course, we want to improve formula to make more and more breast milk-like. And I particularly worked on a compound called alpha-lactalbumin. The story around this, basically, alpha-lactalbumin is the most permanent whey protein in breast milk. It also exists in cow's milk, so the base of what formula is basically, but a much reduced content. And therefore, we saw, well, this protein or this peptide is there in breast milk. What happens actually if we improve the protein content with this predominant protein from cow's milk, but in infant formula as well? And we can actually see that if you do this, you can lower the protein content, because alpha-lactalbumin is a major source of cysteine and tryptophan, and therefore we can reduce that overall protein content of formula, and therefore, of course, we don't have those problems, because everybody knows if you feed formula to your baby, they're slightly more pudgy, they gain much more weight than infants fed breast milk. And that's actually the added protein content to make up these lack of amino acids in formula. And if we just add this peptide in formula, we can reduce the overall protein content, and babies become much more leaner, much more similar to infants that have fed human milk. But also we saw, because tryptophan is an agonist, an opioid agonist, we can actually see that this actually improves sleep patterns as well. And of course, we can see sort of like infants fed formula, maybe they're more prone to be more sleep deprived, maybe they're a bit more cranky, maybe even colic as well. We could see that by adding just this alpha-lactalbumin to this formula, we had all these problems reduced. But also we did another study, because you have to go back, I'm a microbiologist, I look at infant diseases as well. We actually did a study where we looked at enteropathogenic E. coli. And so enteropathogenic E. coli are the major source of bacterial pathogens in formula for infants that don't have a good water source. Okay. And if we take this alpha-lactalbumin in this milk, of course, then we saw that, well, even if we have enteropathogenic E. coli in there, the diarrhea disappeared. Compared to regular formula, compared to formula with other proteins that we added as well, we had the same results that we saw with breast milk, just by adding this other formula, this other peptide in there.
How powerful. And by just adding this peptide, you were able to do all of this. Now, is the idea then for the nutrition of the infant baby to move more toward adding this type of peptide, lowering the protein, of course, from the formula milk, but extracting more of this from cow's milk? Or where does this go next from here?
Well, I launched a product actually in South America with an added alpha-lactalbumin to the infant formula. And this is, of course, now available on the market. What we see now, of course, you see this trend of other compounds that are found in human milk, and actually humanizing formula by adding something called, human milk oligosaccharides. And we can see this is like, so that has been the buzzword almost for the last few years actually, in the infant formula market, where contains human milk oligosaccharide, therefore becomes more human-like, that formula. But we have so many other compounds that we can add as well. We can have DHA, which is also common as well. We can have prebiotics like GOS, galacto-oligosaccharides in there, and other contents which all improve the overall composition of formula and make it more and more breast milk-like. We still can't mimic, of course, what we can see in human milks by adding immunoglobulins and other things, because breast milk, of course, is a highly dynamic compound or product that changes according to the needs of the baby. And we can't achieve this, of course, that sort of focus or day-to-day shifts or even intraday shifts with breast milk. But we can improve, certainly. We are certainly much more improved than what we saw than when I grew up. basically. I was given formula when I was a baby, and if I would be growing up now, basically, with what is available, of course, maybe I would be completely different. Who knows?
How interesting. You know, a moment ago you mentioned even that connection between infant nutrition and then a future state and like down the road, perhaps even the onset of a chronic condition. Can you explain to us what that relationship looks like and how it translates back to infant nutrition? And then what key insights do you have from this field that might even better our understanding of this relationship between diet and genetics? Essentially, how much of it is in our control and how much is completely out of our control.
I would say, unfortunately, maybe it's in the worst case completely out of our control because you can see we're 1.3 to 10 times more bacteria than we're human. If you look at the cells in our body, basically, we're also many more times in terms of genetic actually bacteria than we're human because bacteria outnumber our genes many fold, of course. And therefore we can say, okay, well, if we have all this genetic potential, if we have all these different cells laying around us, basically, of course, it will have some sort of influence. And therefore we can say, of course, what we see now is that, well, maybe some of those chronic diseases start in the gut. And one of the big thoughts right now is something called low-grade inflammation. So the thought is we don't have the right diet, we don't feed the right bacteria, and therefore their signals and compounds are being excreted by those organisms that grow up based on what we feed them. And that these signals that come through may it actually cause leakage in the gut, and these signals come into our body, into other places, start a very low grade inflammation in the gut, basically, which sort of cascades onwards and spirals out of control to where we have cardiovascular disease, metabolic disease, or any of these other chronic diseases, which are highly prevalent, of course, in our society these days.
Oh,
But
interesting. Uh, oh,
again,
please go ahead.
I would just have to say that um, a lot of these things, there's a lot of research going out, but of course, sometimes research is a bit contrary to each other. What we do need is more larger scale clinical trials for this with a good definition of what the outcome is on those clinical trials. And this is something that's still lacking. We're still going with this. And of course, part of this application that we're trying to submit is trying to improve this immediate need of providing a diet that's, first of all, sustainable for the planet, but also sustainable for humans. And sustainability for humans, of course, improving these non communicable diseases with those immediate needs, and then, of course, enlarging this to all of society in the end.
You know, speaking of which, among your current projects, what are some of these exciting discoveries that you're coming across that you've made that could really change how we think about food and nutrition, and again, impacting this relation with chronic conditions down the road?
Well, of course, most of my research is still with infants because that's really my passion, of course. And we see in my research, we've done research on different formulas, well, in clinical trials where we looked at different compositions of breast milk, of milk formula, I should say, at this point, of course. And we do now follow more infants, of course, in a longer period of time to see how they actually grow up with these different formula. And this is again something that's ongoing, but of course we can see that some of the composition and some of those compounds we put into breast milk do have a significant effect in terms of microbiota, but maybe also, of course, chronic disease in the future. This is something that goes on with infants, but of course we can take this knowledge as well um, into humans, well, into adults. Of course, babies are humans as well, of course. Um, but we can see, of course, for some of the troubles that, of course, now these days as well, there's a lot of plant-based nutrition that is coming up. Of course, we all want to do our part to improve life on the planet for the next generations to come. And plant-based nutrition, plant-based proteins in particular, are a big part of this. Um, what we do maybe sometimes see is there's maybe some anti-nutrients in plant-based proteins or plant-based products that would need to reduce. Maybe some of those plant-based proteins as well aren't as good with bioavailability as some maybe the animal counterparts. Um, this can be, of course, also maybe aided through different extraction techniques, maybe also through fermentation with bacteria. We can take down some of those compounds that we don't want to have and improve digestibility as well. And of course, if we do that, then of course we have an effect also on the gut and maybe aid some of this low-grade inflammation as well and improve the chronic diseases that are going on as well.
And you bring up such an interesting point around sustainability and sort of looking ahead at some of these challenges. What are some other promising, sustainable, alternative protein sources you're exploring in your research, and how might they impact our approach to nutrition today?
Well, of course, first of all, I mean, everybody looks at plant-based nutrition right now. And plant-based proteins are by far the biggest field in that. Um, Certainly, we're doing some activities with that, but also some of the other compounds, we're looking at single cell protein maybe as well. Of course, fungal protein is a big part of this. Everybody has heard of corn, for example, that has been around for a long time already. And so some of these proteins that aren't maybe even available on the market yet, um, that maybe we haven't even thought of as well. Also, algae protein is a big thing that's coming up as well for the recent years as well. And certainly some of these algae, we're having very few algae right now that are on the market. Everybody knows about spirulina, chlorella maybe, but there's of course thousands of different algae that nobody has looked at, and maybe those are the sustainable protein of the future. But just nobody has looked at this yet. And this is part of the activities that we are going have ongoing as well to look at different algae. What are the protein contents? What are also the other products in this algae as well? Of course, everybody knows algae oil as well, so omega-3 fatty acids, proteins, but maybe also the exopolysaccharides. So there we go back into prebiotics as well. So we can maybe split that cell and take its parts and make the best out of the separate parts of the cell, basically, and then provide an alternative. Of course, algae can be a carbon sink, so we take some of the carbon dioxide back out of the atmosphere, and therefore we're creating food that is getting more and more carbon neutral, or even maybe taking some of that carbon dioxide out of the atmosphere. So we're improving by eating more of these new diets, basically, we can improve the planet as a whole as well.
How interesting. How does as you mentioned that, how does your research transform food waste into valuable bioactive compounds? And what potential benefits does this offer for both nutrition and environment sustainability?
Well, of course, we always see, I mean, going back to a plant-based protein, we can say, okay, we have these proteins in there, but how much protein is in a plant? It varies quite a lot, of course, but um, not maybe strictly algae, but not strictly plants, but maybe algae, maybe have a 60% protein content if we really sort of like grow them perfectly and um, are very sort of like positive in terms of like protein content. But of course, that means there's 40% other things in there. And we're now living in a stage in an environment where we want to have a circular bioeconomy, it's called. So we're taking a product and we're taking everything else that would be usually thrown away and putting that into different product streams as well. So we have a net zero waste that's being produced. So every single compound that is being produced within a product stream gets used for different product streams to the point where you have nearly no garbage left, basically. So no surplus food waste in the end. Another project for this, of course, also maybe taking at some of these foods that we use not right now and throw them away as well, because a lot of food is being thrown away. I think in Switzerland alone, it's 10 tons of bread per day that gets thrown. And taking some of those waste that are generated, of course, and putting those into different food streams. And traditionally, of course, we've done sort of waste valorization, as it's called, for a long time already. But we were looking at really sort of a low monetary value for those waste streams. We can put them to biogas, we can put them to animal feeds, and of course, animals, 1,000 liter of water for 1 kilogram of beef is incredibly wasteful, and that's why we go into the plant-based products, of course. And therefore, of course, we're trying to just see what we can do with these different compounds that we generate that would be considered waste traditionally. And we're trying to find solutions, whether it's a food, whether it's maybe food packaging. So, for example, we had a project on where we had crab shells or crabs basically in the production back in Ireland. And then we took the crab shell. Of course, the shell gets thrown away, and usually the shell will be thrown back into the ocean. That actually causes sort of like oxygen depletion in the ocean and dead zones in the ocean. So now people were putting it into compost heaps, which also cause sort of like big troubles on land. So, but if we take the crab shell in the end, then we can actually make biodegradable plastics out of that through comes something called chitin and chitin hydrocolloids. And then we can basically say, well, we take the crab meat and we wrap it again in the crab shell. But we just transformed the crab shell, which we consider meat or waste, into a packaging that can be bought and then thrown away, and eventually it's degraded. Or maybe we can recycle the packaging as well. That would be the next step.
How fascinating. I think, Dr. Barker, just talking to you, I can see like there are all of these different opportunities. And going back to what you mentioned earlier, is like we're only really limited by the research that we have at hand because there's so
Absolutely.
many different possibilities in the way that a lot of these things can be broken down, whether within our nutrition, packaging, food waste, all of these different challenges can really be through innovation, through this type of research that you are doing. We can find different pathways to move closer toward a sustainable lifestyle, right?
Absolutely.
What a fascinating conversation. I know we're toward the end of this episode, however, Dr. Brooke, at this time, and I'm sure there are people listening going, oh, I want to learn more. So for them, how can our listeners really connect with you and continue to learn about this incredible work that you are doing?
Well, they can go through my LinkedIn page, for example, or through my webpage at the university, which is available. So you just have to look up my name basically, and I should be, well, there's not too many Wolfram Brooks around, so I'm sure you can find me quite easily, I would say. But there's certainly many options. And of course, always, I'm always happy to be contacted by people and talk about the research or what is possible because we're certainly only limited by our imagination in the end.
Oh my. On that note, Dr. Brooke, thank you so very much for your time. To our
You're
listeners,
quite
thank you
welcome.
for tuning into another episode of the Yumlish podcast. At this time, head over to our social media and answer this quick question. How would you reduce food waste at home for yourself? Hopefully, this episode inspired you to take some actionable steps. Again, head over to our social media, go to @Yumlish on Facebook, on Instagram, find this podcast post, and comment below again to tell us how would you reduce food waste in your home. Dr. Brooke, thank you so very much again for your time.
It was an absolute pleasure.